Okay, so let's start. So hello and welcome to everyone. So I'm here today to represent the GDR Imabio Industrial Partners Club. This club is made up of around 20 private companies who support us, the GDR, throughout the year at the different events that we organized, and including, of course, Imabio, but not only. And so the club also organized some specific events dedicated to the companies. For example, the TED Imabio that we organize also once a year. And today it's a great pleasure to start this new series of video conference. So it will be a regular event every first Tuesday of the month at 1 o'clock. So except for the first one, it's not the first Tuesday on the month. And so you will all receive the information by the email address of the GDR Imabio, also on the GDR Imabio Twitter account. For all people who are not members, of course, you will have also the information of our website. So the format that we decided with the industrial is that the invited company will have a few minutes to present the technology it wishes to highlight today, followed by a scientific presentation in a variety of large biological fields. And this academic is invited at the suggestion of the industrial company. So they propose and they invite this academic. So after this time of presentation, you will be invited to ask the questions to the two speakers that you want to ask. And I would also like to inform you that this video conference is being recorded and will be accessible on the GDR Imabio YouTube channel. So you will find all the links into the invitation that you have received. So today I would like to thank Abberior, who is the first industrial partner who has agreed to start this series. And I also thank Cyril Favard, who is a member of the GDR Imabio steering committee, who accepted to moderate this first video conference. So enjoy, and Cyril, it's your time.
Okay, thank you, Virginie. Thank you very much. So it is our great pleasure to welcome Jakub Chojnacki and Frédéric Eghiaian for this first webinar series of the series to come of the Imabio Consortium. So I'll make a brief introduction to Jakub's work. So Jakub did his PhD at the Johnson Max Lab in Australia, and then moved to Heidelberg to start his first postdoc within the team of Klaus Lietz and Barbara Müller, where he studied HIV, and he used a single molecule localization microscopy to make super resolution images of HIV. And then he moved to the University of Oxford to join Christian Eghiaian as a second postdoc, and he switched to STED and became an expert of STED and associated technologies such as fluorescence correlation spectroscopy and scanning fluorescence correlation spectroscopy that he mainly applied to HIV study. Now he's a research scientist at the IrsiCaixa in Barcelona, IrsiCaixa AIDS Research Institute, and he's also at the same time the microscopy facility managers at the IGTP. I make it short, Jakub. And it will give us insight of how much useful is the super resolution imaging in understanding the HIV membrane dynamics. And according to what Virginie said, before I start talking about the science per se, Frédéric will make a short introduction to the technology, the STED technology that is currently mainly distributed by our partner, Abberior. So Frédéric, it's your time to talk. Thank you very much.
Thank you so much, Cyril, for the kind introduction, Virginie, for the invitation to that brilliant scientific event, and Jakub, thank you so much for participating in this. So I will be sharing now a short overview. Okay, can everybody see the presentation now?
Yes.
Excellent, and you can all hear me. Yeah. Very good. So Abberior Instruments, I will introduce you. The company briefly is specialized in confocal and super resolution techniques, namely STED and mean flux microscopy. And we aim at providing solutions that range from imaging of whole tissues to single molecules. And we really want to stretch the application range of nanoscopy with our instruments. It is actually a twofold company, and I have the pleasure of giving this talk from Göttingen, from the headquarters of Abberior Instruments right now. So right next door, there's a staircase, and you can see that the company is split in two. There is the Dyes and Labels, which is a separate company that produces dyes for all purposes, but particularly specialized for super resolution microscopy as well. And Abberior Instruments, to which I belong and for which I represent Abberior in France, is specialized in super resolution and confocal. The company was founded more than 10 years ago, almost 11 years now, and it's been growing very nicely. I think that the timeline that you see here shows the main motto of the company, which is R&D. It is a very R&D friendly company that stems from the laboratory of Stefan Hell, who was awarded the Nobel Prize for the invention of STED, together with Ed Bedzig for Palm and W. Murner for single molecule in fluorescence. And so this company was founded by Stefan Hell and his colleagues, and with the idea of transferring the technology and disseminating cutting-edge super resolution and confocal microscopy. And you can see the timeline of the developments. We offer 4 types of instruments, 3 of which are STED microscopes, and I just make a short word on mean flux, which is the latest addition to our portfolio of instruments, which is the highest resolving 3D single molecule localization microscope on the market, and also the fastest single molecule tracker. And you've probably seen the recent science publications on tracking of kinesin in vitro and in living cells. So why super resolution? It's because, as you know, all microscopies, optical microscopies, and in particular fluorescence microscopies, are subjected to a diffraction barrier. So what is the diffraction barrier? So when you pass a collimated beam through a convergent lens, then the beam are not focusing into points, but they are going to cause a diffraction figure, which order 0. Here is going to be a spot. And the spot has elongation in a plane, which is directly proportional to the wavelength of the light that we are using. And this is divided also by the numerical aperture of the objective. So you can see how the resolution of the microscopy systems are related to the physical property of the instruments, be it the numerical aperture and the wavelength of light. So to study biological species, as you will see from Jakub's application, it's very important to, especially if you're interested in molecular mechanism, it's very important to dip inside the molecular, structural, molecular arrangement and molecular dynamics at the scale of molecule or supramolecular complexes. And you can see here immediately that be it single globular proteins or cytoskeletal fibers, chromatin, eukaryotic or even prokaryotic ribosomes, or viruses are all below the resolution range of usual fluorescence microscope, beam and epifluorescence or confocals. So the stimulated emission depletion, which is STED microscopy that Jakub will talk about, is based on confocal excitation. And as we excite the fluorochromes, they are brought to a higher energy state, an excited state. And by emission of a fluorescent photon. By applying a redshift in the shape of a donut, what we do is that we bring all the fluorochromes in the periphery of the donut back to the ground state without any fluorescence, but by a process of stimulated emission. And this leaves only the fluorochromes at the central region as capable of emitting fluorescence. And as we increase the power of a STED beam, say, on those 20 nanometer fluorescent particles, then what happens is that we increase the resolution until we reach the point where we see the 20 nanometer particles as they are, that is, as 20 nanometer objects in plane. So that is pretty much the best performance of our instruments in 2D STED at the moment. But this can be also applied to biological samples. You will see it in a minute, but here is an example, a very famous one, on nuclear pore complexes labeled in 2 colors. And this changes a lot the situation from the confocal microscopy, where we can surmise that there is colocalization of a green and red color, but we cannot get a deep structural information on the arrangement of the nuclear pore in those 2 colors. Now, we can go further than 2D STED. We can also increase the resolution, not just in plane, but also in all 3 dimensions, and yield an image with isotropic resolution, which is not the case of confocal microscopy. As you can see here, a confocal spot is going to be very elongated out of plane. Whereas we have a 200 to 400 nanometer extent in plane, then the Z extent of the confocal spot is going to be in the range of 500 to 700 nanometers. And in that case, the 3D STED gives us an excitation volume and of about 75 nanometers in all 3 dimensions. And so we get a resolution, final resolution of about 75 nanometers in all 3 dimensions, which is very useful to study morphological aspects of things. And if I just have 30 more seconds, I would just like to say that Abberior, besides commercializing STED in 2D and 3D, as well as confocal, is trying to push the limits of all those modalities in depth, live imaging, also trying to improve the signal to background and also resolution. So I would just like to show an example on how we push the depth of super resolution and confocal imaging. What we do is that we use a modality which is called adaptive optics, which is based on the deformable mirror placed before the scanner of the instrument. And this way, we are able to bring 3D STED in depth that were not achievable previously. So you can see here on that cleared kidney slice labeled in 2 colors on podocin and nephrine, the result of 3D STED uncorrected and 3D STED corrected with adaptive optics, where we can get distant 3D STED all the way down to 80 micron depth. And that's it for me. I'm very glad to now leave the stage to Jakub.
Right. Thank you very much. Let me share the screen then. Let me now this moment, please. Sure. I hope everyone can see my presentation right now. Yep.
Yeah, perfect.
Okay, fantastic. All right. Well, thank you very much, and for, of course, for the opportunity to give this talk on this webinar over here. And the story that I'll present to you is revolves around application of STED microscopy towards HIV research. The story, for the disclaimer, the story isn't the newest anymore.
La première slide de la première truc. J'ai dû louper l'intro.
Sorry.
Okay. Everything all right? Okay, so the story is not the newest anymore, but although the last publication for this story that I'm telling ended in 2021, was published in 2021, but I think what it nicely illustrates is application of super-resolution microscopy to, in this case, a viral research. Not only HIV, but also the results and general techniques that I'm showing here are very much applicable to a lot of other infectious agents. So to make a quick overview, I am going to, the presentation outline will be as follows. I'll first give a very brief overview of STED, which Frédéric has already actually put in more detail. But then I'm going to talk about 3 aspects of basically 3 stories in short, which one builds upon the next one. So I'm going to talk about HIV and distribution during assembly and entry, HIV surface ENV mobility study, and then finally lipid and ENV mobility at fully infectious HIV assembly sites. So a brief overview of STED super-resolution microscopy. This is pretty much what Frédéric said just before. The resolution of a conventional fluorescence microscope is limited. 250 nanometers in focal plane and 600 nanometers along the optical axis in confocal microscopy will give you the image as you see below for the nuclear pores as was shown before, but I also show a HIV image. This is how a single HIV virus looks like when looked at a confocal microscope. The blue circle represents the actual dimension of the HIV virus particle, and yet we see a diffraction limited blurry spot. In this case, this is end surface molecules on the surface of the virus. And then, of course, with stimulated emission depletion microscopy, which, as Frédéric said, you apply additional depletion beam to engineer the point spread function to reduce the detection volume during the imaging, will give you results like this, where for the nuclear pores, like was shown before, you start seeing the structures. And as for HIV, in this case, the green signal is still diffraction limited, but the orange signal becomes you can actually see the distribution of individual surface molecules on the surface of the individual HIV virus particle. And just as a comment, the STED microscopy, at least in my mind, represents a current generation of superposition techniques, which roughly oscillate between the resolution improvement down to between 10 to 100 nanometers. Of course, like Frédéric even mentioned, there is already a new generation of microscopy, superposition microscopy techniques that are coming, but that is not the part of this talk. So, going to the general comment on the superposition microscopy studies of HIV replication cycle. Of course, HIV is perhaps the most studied pathogen by superposition microscopy. This is because, of course, the field has an advantage of many reagents. The field is very big. So it was naturally one of the first virus particles that were put under then in 2010 coming newly shown superposition microscopy techniques. And actually, you know, from the overview of the field, basically the studies that have been done really that they are utilizing superposition microscopy have really revealed the previously unknown mechanisms that influence HIV assembly and HIV entry. So this reveals the enormous potential of this application of these techniques in general in study virus replication cycle and virus cell interactions. So HIV is a very good poster child of what can you achieve utilizing these techniques. And like I said before, they're very much applicable to other viruses as well. So to start off the story, I will kick off with looking at the HIV ENV distribution. And this story exactly started in 2007, 2008. So there we started really without knowing anything about whether the superposition microscopy work at all on HIV particles. But the bottom line, the bottom information that we had, the information in the background we had, that we knew that HIV particles incorporate quite a little ENV that was theoretically possible. And also we knew that HIV ENV incorporation, ENV in red, is influenced by underlying GAG lattice, specifically by its interaction with a domain of GAG. Also, we knew already from early studies of electron microscopy that ENV distribution may play a role in HIV entry. And also, the ENV distribution itself may be influenced by interactions with GAG and thus affect the entry competence of the virus. This is because immature virus particles, which have a very ordered GAG lattice underneath, are basically entry incompetent. Also, in fact, non-infective particles, whereas the fully mature virus, characterized by the cone structure, which we all know, is fully entry competent. So we set with the aims, firstly, to visualize HIV ENV distribution using superposition microscopy and establish its effects or effects of GAG processing of virus maturation on ENV distribution, and observe any possible ENV rearrangements during the receptor interactions in wild-type and mutant particles. So, as a snapshot of the results, we were able to visualize HIV ENV distribution in the context of individual virus particles utilizing STED microscopy. And we looked at both mature virus particles and immature virus particles. And then we quantified the ENV class distribution, and we found out that ENV distribution on immature virus particles is very different, much more distributed, broadly distributed than on mature virus particles, in which it predominantly becomes a single cluster only. Furthermore, we actually were able to visualize utilizing a multicolor STED microscopy, colocalization of these ENV clusters with CD receptors. So here we have a T cell, which we zoomed into an individual virus particle interacting with CD4, which is in blue, with orange being a single ENV cluster and green representing individual virus particle. And we're able to do this on several different types of different mutants for HIV. I'm not going to go into too much details, just want to show the potential of the technology and applications. But the conclusions of this study where effectively that we found out basically a novel inside-out signaling for the HIV maturation, where the maturation of internal virus structures causes changes on the surface. And also, we were able to show that ENV clustering on mature HIV enables for efficient virus entry. Actually, this is very important. This clustering is important given how little, around 7 envelope molecules are present on the surface of the virus on average. So this was actually quite a new mechanism discovered back then in 2012. Then we kicked the story a little bit up the notch and wanted to look at utilize STED microscopy to something a little bit more, wanted to be a little bit more ambitious. So we looked at then dynamic properties of HIV on the surface of the virus particle. We knew that the virus particles changed the distribution of ENV, but we wanted to check what are the dynamic properties during the reorganization of ENV that occurs during the maturation. So for that, the aim was to actually look at mobility and dynamic properties on the surface of individual virus particles, and also, of course, to establish whether this maturation status alters the HIV and mobility rates. For this, then we had to actually change, add another technique to STED and basically couple it to fluorescence correlation spectroscopy. And what is that in a nutshell is looking at mobility of the molecules, and we record the fluorescence fluctuation in the observation volume, and then construct correlation curves, which then these correlation curves encode a number of useful information, like number of molecules transitioning through the excitation volume, as well as average transit time. So from the average transit time, we can get molecular diffusion coefficient and molecular mobility of the molecules in question. Here, of course, we combine it with the STED microscopy, where, of course, the standard FCS is diffraction limited. So if you want to study a virus particle, this limited spatial resolution will not be very useful. That's why when we combine it with STED to form STED-FCS, we can now observe things on the subdiffraction volume under 100 nanometers, which is the size of the virus. Here we had to actually, just to make the story a little bit more complex, we had to apply a special modality of STED-FCS, which is called scanning STED-FCS. This is because we had to actually, rather than focus a point on a single virus particle and all the time observe it, due to the very limited amount of fluorophores that are present on the surface of the virus, you would bleach the sample very, very quickly. So we had to apply so-called scanning STED-FCS, in which we scan in line, and only occasionally at very specific intervals we check the mobility of the ENV molecules on the surface of the virus. This is all in separate diffraction mode. An advantage of this scanning STED-FCS approach is that it allows the fluorescent molecules to relax a little bit, and thus limits the photobleaching that occurs in the context of this single virus particle. So here, how this works, we get an additional spatial component to STED-FCS, and we record the intensity fluctuations in the y-axis, and the x-axis is the scan positions. And then we autocorrelate this fluorescence fluctuations in the areas that are representing a virus particle. So here is a picture of the virus particles immobilized on a cover slip. The line represents where we scan. We're using our scanning STED-FCS approach, and we construct basically, this is the intensity carpet, which we correlate with the virus is, and this is the ENV fluctuations of ENV molecules, which are on the surface of the virus particle at resolution of 55 nanometers, which is well below the size of the virus, which is around 140 nanometers. And then, of course, we construct the correlation curves, and we derive molecular data on molecular mobility of ENV per single virus particle. And then we acquired the data on numerous virus particles, and we arrived at the following results. We found out that there is a diffusion coefficient, but for those of you that know, it's actually very, very slow diffusion coefficient. However slow the mature virus particles were, we found that effectively in immature virus particles, this is where the limits were reached of the useful interpretation of the data. We found out that the ENV molecules on the surface effectively almost stop, because this is pretty much the same result as you get when you have a fixed virus particles. Then we actually looked at whether this is actually intrinsic property of the virus particle, or is it something related to the ENV itself. So here we actually turned into looking at a very small molecule, which is completely much smaller than ENV. We looked at GPI SNAP molecule. We looked at it first in the cells, and we found out that in a standard FCS application, it has a diffusion coefficient, which is representative of how you expect of a small molecule like this. However, when we pseudotyped the virus particles with this very small molecule, we found out that even this tiny molecule, which is much smaller than bulky ENV, also has a huge decrease in the mobility, irrespective whether it's on mature or immature virus particles. This is because this small molecule is completely independent of what is underneath, what is the GAG and the nature of the GAG lattice underneath. So to conclude from this study, utilizing STED microscopy and combining it with fluorescence coloration spectroscopy, we have found out that HIV-Env exhibits very low surface mobility rates that are at least order of the magnitude slower than what you expect on the cell membranes. So virus maturation status also was found to influence ENV mobility. And the bottom line of this study was that virus envelope is intrinsically very low mobility environment. And there are several reasons for this. This is because of very high lipid order, extreme membrane curvature, and tight protein packing. I guess at the time when we published, when we're doing these studies, we're all influenced by the virus cartoons like this, where you think that there is a lot of empty space in the virus particles, or whether they do it 2D or in 3D. However, the reality is that a virus particle is extremely packed with proteins and atom molecules. So it's really not a surprise that it's given the dense curvature of the membrane, as well as the extreme amount of proteins inside of it, that the whole environment is very, very different to what you encounter in the relatively large in comparison environment of the cell. So once again, this was very interesting study, pushing the envelope with, really discovering new aspects of HIV. But then we went a little bit further. We wanted to look at the lipid and ENV mobility at the site of fully infectious HIV assembly sites. And this, in the background, is we knew that HIV membrane is enriched in single myelin cholesterol. And at a time when we were doing this study, consensus was that virus assembly and budding occurs at the pre-existing lipid raft domains or liquid ordered domains. However, we wanted to test the hypothesis that instead of targeting pre-existing domains, assembling virus particles may create their own unique lipid environment as it assembles on a surface of the infected cells. So here we actually utilized both approach for fully infectious virus cells infected with a fully infectious virus, but also we did this study in the synthetic GAG, in the synthetic membranes and utilizing a GAG molecules. But here I'll just talk about the fully infectious virus particles. So basically here is the image of the fully infectious cell that is infected with the fully infectious virus. And after 20 minutes, you start seeing a virus budding sites appear at 37 degrees. And then we focus the microscopy, the imaging on those virus budding sites. And here, like before, we utilize scanning STED-FCS approach, except in this time, instead of a line, we used the orbital approach. And we looked at the mobility of the molecules, in this case, actually both lipids and proteins, both inside and outside of virus assembly site. So like I was saying before, scanning STED-FCS approach gives you additional spatial component in which we can look at both what's happening inside the virus particle assembly site and just slightly outside of it. So here we have once again the intensity carpets, and we looked at the fluctuation properties of the lipids and proteins, and we looked at it in different spatial positions inside the virus particle assembly site and just slightly outside the virus particle assembly site. So this is yet another evolution of this principle to give you more information from a single scan. You look at the different positions and you get dynamic properties of the molecules on these positions. So this is a fairly dense slide. So yeah, I'll just summarize it. We looked at several different lipids, and this actually this study has been done in collaboration with Cyril's and Delphine's group. So here we looked at the PIP2, we looked at cholesterol, sphingomyelin, phosphatidylethiolamine, and we found out that both cholesterol and PIP2 seem to be very, very much trapped on the sites of the nascent virus particles at the very beginning of the virus assembly, but sphingomyelin and phosphatidylethiolamine was not. Then I did a small study to confirm that this is also happening with ENV, that ENV also becomes trapped at the inside assembly sites, and it's actually relatively free to move around outside assembly sites. So yeah, so basically to conclude this study, we found out that during the budding, HIV creates its own lipid-specific environment by selectively restricting lipids at virus assembly sites. And this environment, combined with immature gliadin interactions, leads to trapping of ENV and its incorporation into nascent variants. And of course, there have been then since follow-up studies which try to explain why looking at this immature gliadin interactions, why there is so little ENV trapping on the virus assembly site. But it seems like this very specific environment created by the virus is one of the strongly contributing factors of why so few ENV molecules are trapped on the surface of individual virus particles, which then bud out, and you have only, like I was mentioning, only 7 ENV molecules on the surface. And with that, I will actually stop here. And with the take-home message is that the super-resolution microscopy, in this case, STED microscopy, is a very powerful tool for studying pathogens with potential to make high-impact discoveries. And also, with the current availability in general of commercial super-resolution microscopes, these techniques can be easily utilized by a large number of research groups. Right now, have matured very much, and they are very, very user-friendly. So this the sort of first generation of super-resolution microscopy techniques, STED, especially STED, is now very, very mature and ready to use and ready to be applied. When I started this story, we were still very much in the dark about the techniques when the support from the companies and the support of the microscopes was not as high as it is now. So it's really now a good time to utilize these techniques for other pathogens, for example. And I will also finish just by mentioning a little plug here that that we actually in Barcelona, we now recently opened a super-resolution microscopy facility in the high-level biosafety facility. So we do have an Abberior instrument inside high-level biosafety facility where we are aiming to do live cell imaging on both pathogen, fully infectious pathogen research, and animal tissue imaging, because actually this facility also has animals next door to us. So in NCB facility, and you can study a whole lot of different pathogens. So the pathogens that we're working on right now are HIV, SARS-CoV-2, and others. The facility is a service-oriented facility, but you can ask me if you want to about possibilities of of the academic collaborations, especially in the HIV field. So with that, I will stop, and I will thank. There is a lot to thank of my previous positions at Heidelberg University and Oxford University, also Cyril and Delphine's group, which were my collaborators before, and right now the group which I'm associated with, IrsiCaixa, which I'm doing some HIV studies still. So with that, if you have any questions, you can of course contact me on via email here. And now I will take any questions that you may have. Thank you very much.
Thank you very much, Jakub. So if there are any questions, what I propose to make it easy is that you take the reaction button, which is somewhere on the interface of the software, and there is something which is called raise the hand. So if you have question, you can raise the hand, and then I will give you an opportunity to ask your question. Because if everybody activates his microphone at the same time, it's going to be a mess. So yeah. So the name I see is Brolo. So yeah, thank you.
Yeah, thank you very much for this very nice presentation and all the works on the STED and the entry and assembly. So thank you for this. It's impressive, I have to say. And my first question is, which kind of viruses did you use? So is it X4 or R5 viruses? And the second one is that is there any difference between the different viruses, X4 or R5? And also, is there any difference between different viruses within the R5 group, for example, the founders viruses and the lead viruses? So it's something that may influence the entry. So it's the question I have. Yeah.
Sure, that is actually very good question. The initial studies, the initial non-infectious studies, were done on a pseudophyte. There were pseudotype particles, or actually replication incompetent viruses. Okay. However, the viruses in the fully infectious studies were using a T-cell specific virus, which I think is R5. And just to answer your question about founders viruses and the differences, this is unfortunate, this is something that I did not have the time to study exactly, but I have, now actually I have a group which I'm collaborating with. We actually precisely asked this question: can we look at the founder viruses? And even in Oxford, this question was floated, but I just never got a chance to see the differences. So right now, my answer to the question is, I don't know, but hopefully we'll soon find out, because there are some clues to indicate, especially from the studies of founder, how they are very different to basically ladder adapted strains. So I don't know, but hopefully we'll find out.
Okay, thanks.
Okay. So before other questions come, sorry, I'm going to ask one, Jakub.
Sure.
It's more asking for a comment than a question per se, indeed. So the mobility of the envelope is very, very, very low,
Exactly.
whether it's mature or immature. And,
yes.
And the question I was wondering is that, is it fast enough as a mobility to make sure that or is it maybe a trick that uses the virus so that once you have targeted the receptor, the other one moves so slowly that they will be close to this one and they will facilitate its kind of cooperative system? Because in the scheme you've shown the envelope in one case to be quite separated but not moving, and in the other case to be close to the other ones because they move a little bit faster. So is it just a cartoon, or is it really what you observe?
I think this is just a cartoon, because I mean, what's happening really is when we look at the virus particles, already fully mature virus particles, they already are pre-clustered. So whatever, once the env is released from the GAG, there is, sure, the mobility is slow, but the virus spends, I suppose, indeterminate, amount of time in a solution or whatnot. It will have enough time to pre-cluster, especially since there are self-association domains within env itself. So even though it's slow, I think, even if it waits, we never actually tried to do any targeted live cell imaging on release and see how quickly those molecules would cluster. So release the gag lock, so to speak, and then look live time-lapse microscopy on how individual virus particles will cluster. So, but I would still suspect, given how small the virus particles are, then they would actually still quite rapidly get into the preferred single clustered distribution. Does that answer the question? I hope I understood right.
Yeah. In some extent it does. Yeah. Okay, thanks, Jack. We have another question.
Sure.
So it's in the discussion folder. So it's very nice. Maybe I missed it from Bruno Baumel. Well, can you be sure that the virus creates its PIP2 call enriched environment and not using a PIP2 call enriched domain to assemble?
Okay, well, that's first of all is because we have information immediately outside of what's happening outside of the virus assembly site, and in there the PIP has a very different behavior than just inside the virus assembly site. Very importantly, the liquid order domains are they have a very different if whatever you call them, they have a very different properties. Molecules there move slower in there. And so we inferred that precisely because there is a very big difference on how PIP behaves just outside our assembly site, then there are no, it's not utilizing a pre-existing domain, that it rather creates its own. Also, sorry, I mean, because as not to confuse myself, I think it's big, we can see that there is a specific, trapping of the lipids, because other like sphingomyelin, which is related, and PE are not trapped in there. So normally when you have a general liquid order domain, I think all the lipids would be trapped. So here we have a very specific environment that is being created. So yeah, I think that's why we inferred that it's not a pre-existing domain.
Are there any other question? Don't be shy. You can ask any question you want.
Sure. Technology, application, or about the virus. Because, I mean, the point of this is, of course, to also comment on the application itself. The latest studies, of course, were much easier than in 2010, thanks to advances in instrumentation. Still challenging from the virus point of view, but definitely a bit easier from technical point of view.
Okay, I'll ask another one before somebody I'm going back again to this low mobility of envelope. Do you think you could have imagined, for example, to use something more like imaging correlation spectroscopy instead of just scanning correlation? Because when you scan, you get there from time to time, and then you collect a given amount of photons, and of course you can self-correlate this within time. But when it diffuses very slow, I would say that FCS start to become not as appropriate as imaging, for example, FCS, imaging correlation spectroscopy will be. So did you try to do that?
Yeah, that's a very good, that's a very, very good question. In retrospect, precisely this is what we should have done. In the end, we actually didn't do it because we didn't have the instrumentation, I think fast enough cameras for the image correlation spectroscopy on there. But no, irrespective, we actually haven't tried that. And by then I left the laboratory. So this is something that that could be addressed precisely, like I was saying during the presentation, that there's such a slow mobility is what it precisely push the FCS and the data to the limit, and you would probably have to use slower techniques which can address slower mobilities there, like image correlation spectroscopy. I totally agree with you, but the answer is no. We actually never tried with ICS there. So yeah, something yet another avenue that just myself I'd never explored. There's so many different branching avenues that I could have explored, but I didn't.
Yeah, but we can't do everything at the same time.
Precisely.
Frédéric, you have a question?
No, I don't have a question. I have a comment because I realized that I said something a bit incorrect. So when I was describing the 3D STED, I was saying something about the excitation. So the 75 by 75 by 75 volume is a fluorescence emission volume. So the excitation is confocal, then it's depleted, and it leaves emission volume of 75 nanometer cube, so to speak. That's all.
Okay, thanks. So if there are no other questions, I think we will end up here this first webinar. So thank you very much, Jakub, for giving us and sharing us your knowledge on this technology and its application to viral assembly.
No
Thanks
problem.
also,
Frédéric.
so somebody comes in. A5 Science. I don't know who is. If you have the question. Oh, no, it's a clap. Sorry, it's not. Oh, yeah, it's okay. Thank you. So I end up like this. Thanks again, Jakub. Thanks, Frédéric, for introducing also the technology. And thanks to Virginie. I think she will close the webinar.
Yes.
And thank you to all for attending.
Thank you very much.
Okay, so thank you all for taking part to this first video conference, and many thanks to Frédéric and Jakub. Thanks a lot. Thanks also to Cyril to moderate this session. And so our next meeting is scheduled for Tuesday, 4 July at 1. It will be with our second partner. It will be confocal.nl. And they have invited the Professor Erwin Peterman from the Vrije University in Amsterdam. So I hope to see you next time, and thanks a lot.
Thank you. Bye-bye.
Thank you.
Thank you so much.